Hello, my name is Luke Beeson, and my lightning talk today is looking at ridgeline biodiversity. More specifically, the question I was pursuing was, does biodiversity change on and off ridgelines? And if it does elevation play a role in this trend? This analysis was also done in conjunction with looking at surveying errors that were potential in the analysis and whether or not it was statistically sound based on the sample size that were used. Some caveats that were done to control for surveying error were that only ridgelines along the Long Trail within the boundaries of the Green Mountain National Forest were taken into account, and only elevations within the Green Mountain National Forest were surveyed to keep data trends as consistent as possible. The data that was used were iNaturalist observations, as well as the Green Mountain National Forest boundaries in southern to central Vermont, and the Long Trail boundaries with a 100-meter buffer. But the Long Trail was only looked at within the bounds of the Green Mountain National Forest. The Long Trail boundary was used to look at ridgeline presence and biodiversity there. The group was manually split into two feature classes: the points where mammals were observed on ridgelines and the points where mammals were observed off of ridgelines. And the elevation analysis yielded over 300 observations, and these were then grouped into 100-meter elevation intervals from 200 meters to 1,000 meters. Anything below 200 meters or above 1,100 meters were excluded from the analysis because these sampling areas were just relatively so small in the region that they were yielding outlier results. These results were then used to compare the number of the observations, the species richness, and the Shannon's diversity index between these groups. Again, these groups were on-ridge and off-ridge observations, and subsequently elevation gradients, which were groups split into 100-meter intervals. From this, two statistical tests were run. On the ridgeline observations, an unpaired t-test was run, and on the elevation data, a ANOVA test was run. Here, Figure 1 shows the number of species observed, the total number of observations, and the Shannon's diversity index for the two groups on-ridge and off-ridge. A statistically significant difference was found here, with less biodiversity in both richness and Shannon's diversity index found off of the ridgelines. This data was corroborated by the fact that a statistically significant difference in observations was found along a elevation gradient as well. Here, the mid-range elevations had more richness and a higher Shannon's diversity index. This was also associated with a higher number of observations, which is a potential source of error. Overall, this data shows that significantly higher biodiversity is found off of ridgelines, which is then supported by mid-elevation ranges with high biodiversity levels. This is also supported by a myriad of different papers showing that increasing elevation brings decreased biodiversity across many different kingdoms. This is plants, animals, fungi, everything. Some potential causes of this trend are that there are fewer resources in high elevation habitats. Also, these habitats have more exposure to the elements and are therefore more energy intensive to survive there. But some potential sources of error from my analysis may be sampling bias and uneven sampling effort because the observations were simply opportunistically sampled and gathered from iNaturalist data. Thank you for your time.